With the seedless vascular plants, one thing I need to, what the heck is going on with that? Why am I having that little pop-up thing? Please move this window away from the shared application. Never saw that message before. What the heck is that about? I don't know. What are they talking about? Zoom, you're killing me. All right, let's just see if it'll behave. Okay. One thing I wanted to point out, you need to know the phylum names. Gotta know them. There's going to be questions on this, several questions, looking at which group of plants belong to this phylum, or what phylum does this group. Those kind of yellow highlights, Lycophyta, Polypodiophyta, Pyrrolophyta, Pseudophyta. There'll be several questions on it. So don't forget to get those committed. Ecosthetophyta to me is really easy. Excellent. For the horses, so that's the horse tail. But then, yeah, the rest you have to come up with some tricks to figure, remember who belongs into which phylum. So that's one thing I wanted to point out. Then, yeah. Let's see. Okay, so let me go back. Let's see. At the top here. So we were looking at after the bryophytes, the moss into the seedless vascular. And so with the seedless vascular plants, I'll be asking about their characteristics, the seedless. All right, so we're doing away with the release of spores idea. Now it's a release, it's still the spore idea, sorry, but this is vascular plants. And so now we're going away from these very petite little bryophytes that have no true vascular tissue, no true roots. And now we're shifting into plants that have vascular tissue. Some have roots and some don't. Some have more like underground stems. But basically, we're seeing the presence of vascular tissue now, but they're still seedless. And so, knowing the general life cycle too, so it's safe to say everything on this exam, the sporophyte dominates. There will be no exception. And so that's a safe one to think about. Just remember sporophyte from this point forward. It's always the sporophyte that is the dominant part of the life cycle. Any questions on the life cycle of the seedless vascular plants? We still have to think about the alternation of generations idea. I guess we got into the whisk fern. So here's an example with the whisk fern. Yes. Why? Me. Yeah. You're looking here. Yeah. Yeah. So they're trying to show that on one gametophyte, you have both male and female. Yeah, most bosses are separate. There's a male one, then there's a female. Yeah, so we can have this situation where they're on the same one. So that, yes, you would say it could be monoecious, one house for both male and female. And we still have to think about the words antheridium, archegonium, or sperm and egg, respectively. And then the fertilization idea gives rise to the zygote. So once the sperm and egg fuse, then you start calling it the zygote. And then once there's significant, cell division and some growth, then they start using the term embryo. But basically that will develop into the baby sporophyte. And that's what you're seeing. Sporophyte is starting to grow, and pretty quickly the sporophyte separates from the gametophyte. They don't stay attached to each other. And then with the sporophyte, the job of the sporophyte is to make spores. And so you're looking for sporangium tissue that will hold the spores. So a lot of the same terminology that we already saw with the bryophytes still showing up here. And let me I'm trying to find something quick here. Okay, here it is. Okay. Just wanted to reinforce the idea that you still are requiring free water with the seedless vascular plant. You need free water to get the sperm delivered to you. So when you're studying, it's a nice idea to think, you're gonna have this slide set open to be learning this, but like keep it open, because then as you shift into the gymnosperms, I'll be asking on the exam, like, okay, what's different? What's different about the gymnosperms compared to what we saw with the seedless vascular? Like trying to get you to think about the evolutionary process and how things shifted, and you get some special adaptation that distinctly separate these groups of plants. Let's see. Let me. What else on this slide set? What content on here do we need to go over? Yes. Yeah, so typically when you hear the word leaf, most of us think about maple leaves or oak leaves. So that's a megaphyll. When I say normal, that's what I'm thinking about, what we normally think about, maple leaves or oak leaves or something like that, the normal leaves. And so they think that the evolution of that occurred through branch tissue, that instead of being in different dimensions of space, all the branches ended up in one plane of space, and then in between the branches it filled in, kind of like duck feet webbing, that kind of idea, where it filled in and became what we call the leaf tissue. So that's the megaphyll. Microphyll, you might not even think about that as a leaf because it just doesn't look like much. Microphylls tend to be pretty petite, often very thin or scaly. Very simple, just a single vein. And then if you, let's see, so with the whisk fern idea, those are enations. So that's like the forerunner of leaves, those enations idea that the whisk ferns have. It's just this little tiny flap. And then from eventually those enations evolved into some plants, evolving that into a microfilm. And then if you look at the chart, I think it's there where they talk about whether there are any leaves or not. Yeah, so for Lycophyta, phyta is that's a microfilm. That's a microfilm. Classified as a microfilm. So really in this group, it's only the true ferns, the Polypodiophyta, that have megaphyls. And you know, most ferns, when you look at them, you say, oh yeah, that's a leaf. Let's see. Any other questions on the seedless vascular plants? Let me go. So for example, with the phylum Lycophyta, you know, the three groups here. Plum moss, brown pine, spike moss, silver. I usually refer to them with their common name on the exam. You know, and so I might say, what phylum do the spike mosses belong to, or the plum moss, the brown pine, not to pick from the multiple choice. Very cute. Seedless vascular plants. Anything else on here? Well, don't forget about the spike mosses. They're weird in the sense that they're the group that is showing this megaspore, microspore. Up until this point, we've only been really talking about spores. Now we have this idea of megaspore, mega is part of the word. Megaspore, about the megaspores will be developing to megaspore. We have female, long female. When you add the microspore, that job, you know, spore's job is to, once it gets released, start to grow and megaspore. A microspore develop into a micromegacyte, which is solely male. And so we're looking for sperm inside of the antheridium. And then once we get into the gymnosperm, this is the strategy. We have the heterosporous condition. This is heterosporous, so there's two different spores. Megaspores are physically larger. Microspores are small. So the seedless vascular plants, it's really only the spike mosses that we see that in, and then all the others are just regular. All the spores are the same; they're homosporous. But when we jump into the gymnosperms, we're going to see this trend continues, where you have megaspores and microspores. Anything else in the seedless vascular plant? Always be the female. Don't forget to learn a little bit about their ecology or their uses. Yes. Yes. The vascular tissue, that's, you know, this group evolved vascular tissue, but it's the more primitive type. So we're really thinking about, we've seen this already way back when we looked at plant anatomy and we were looking at stem tissue, but really what we're zooming into is this group up here, A, B, and then some of the C here. So protostele, This means that you have a solid cylinder of vascular tissue smack in the middle of the stem. And then the saphonosteal idea, where it seems like most species of the Cebus vascular plant will do this. It's a ring of vascular tissue. So they think the protostele is like the most ancient, like that was the first run at vascular tissue. That's what they came up with. And then over time it evolved into a ring of vascular tissue being shown with the saphonosteal, and then also this idea of it's a ring, but there's more like discrete clusters. But I'm trying to show here. With C, that figure C, it's like show discrete clusters, but overall it's a ring pattern. Yes. Yeah, yeah. Similar. Mm-hmm.
Yeah.
Yeah, I do, and I don't know. Because this is zooming in on stem tissue. So as far as evolution of vascular tissue and root tissue, I don't know how much has been done on that. I'm sure someone studied that, and that's a valid, what you just described. Could that have happened? I could see that, but I don't know the answer. Some do, some don't. And so we say there's vascular tissue, but that does not necessarily mean they all have roots. So if you go back to, I think in here, differentiated root stem. Yeah, yeah, yeah. So with the whisk ferns, they're the ones that they can have like underground rhizomes, like underground stems, rhizomes, but they don't seem to have true roots. But the other groups do. Well, should we jump into the gymnosperms and hit some of those? Gymnosperms. So the shift. Now we see that there's a shift. The spores aren't released anymore. So this grabbing the right screen, zoom here. All right, so with the seeds, the second bullet point, the seeds replace the spore as the unit of dispersal. So what that means is that with the seedless vascular plants, they release out the spores. They let those fly away, go and land somewhere, and those spores should germinate and grow into the gametophyte. So the spores were released. Now with the gymnosperms, it's not until you have the baby sporophyte all good to go inside the seed, then that seed will get dispersed. And hopefully that seed will land somewhere and grow back into the, a sporophyte, a mature sporophyte. So very different strategy. So this slide is important to think about for the final exam. This slide and this slide, these two slides, because this is a huge concept that I'm hoping you'll learn, where you have this idea of butterflies, vascular plants, the spores are really brand new. They're on their own. It's like, okay, spores, you're released, you better go and start making sperm and eggs out there in the great wild. But with the gymnosperms and the angiosperms, they protect the spore. It's retained and very much protected. And then it will develop into the gametophyte attached to the sporophyte. It's all protected by the sporophyte tissue. And so that is a great strategy to really protect the precious egg and sperm, and making sure that you have a high chance of success for pollination, fertilization, and growth and development of the baby sporophyte inside the seed. That's a huge shift in how they strategize, trying to keep their species alive and to spread out and adapt into new habitats. Seeds are much tougher than the spores we've been talking about with the other groups. Right? They're very hardy. And so gymnosperms, angiosperms, they're both heterosporous. They'll have megaspores and microspores. And then remember the job of the spores develop into the gametophyte. So a megaspore develops into the megagametophyte, a microspore develops into a microgametophyte. And I need you to know the phyla. I'll be asking about phyla again. Who belongs into which phyla? Only four. 4 in the seedless vascular plant, 4 in the gymnosperm, and I don't ask about phyla with the angiosperms because there's like a ton. So we won't get into that. And let me go, let's see. Weird. All right. How about the life cycle? How you feeling on that? So we need to zoom in to the ovule and think about what's going on with the megaspore. So now we see a big reduction in the number. How many megaspores are we talking about with the gymnosperm? Just 1. So they put all their, now that's another reason why they protect it so well. And I got just 1 here in this particular developing. So you see the megaspore site, what they call the megaspore mother cell, just 1. That megaspore mother cell will go through meiosis. It's being held within the sporangium, not being released, stays in the metasporangium. It goes through meiosis. That 1 cell will produce 4 cells through meiosis, and then we get left with just 1. 1 survives. Functional megaspore survives. And then the job of that functional megaspore now is to grow and develop into the megagametophyte, still tucked away inside the megasporangium. And so the megasporangium never bursts open and releases the spores and sends them off. And the job of the megagametophyte is to make gametes. So in this case, to make egg inside the archegonium. Whoops. So this image is showing that you have a functional megaspore still tucked away inside the megaspore, further protected by the. So that's like the ovule, that whole thing. Say, oh, look at that ovule. And then that functional megaspore, it's got to grow into the megagametophyte. It's got to develop. So it just goes through lots of mitosis. We got that weird free nuclear stage where it looks like a hot mess, but eventually it'll show tissue-like organization, looks like a megagametophyte. Eventually you'll see the archegonia with the egg in each one. So this is a mature ovule because it's showing the megagametocyte that's developed. That functional megaspore that was sitting here in the previous image, now it's developed into the megagametocyte with the archegonia and big pump egg archegonium. And then that's still retained in the megasporangium, and then nicely surrounded by the integument, except for that little micropyle, that opening where hopefully pollen will end up there. So I definitely will be asking about this on the final exam, this process. Pay attention to the figures as well, being able to identify the parts, okay? What else about the ovule? Do you have any questions on that? Can't forget about the pollen. Gymnosperms, they're a little fancy. They have 4 cells that make up a pollen grain. With angiosperms, it's only 2. So angiosperms reduced it. You don't need 2. Right, but the gymnosperms, they came up with the idea of 4 cells. So when you have the pollen cones shedding their pollen, they're in this stage. There's 2 prothallial cells, there's a tube cell, there's a generative cell. Nowhere in there do we say there's sperm showing up yet. All right, so it's immature. We say that the pollen grains are immature because of that. But then eventually, once you get the pollen delivered to the ovule in a female cone, then eventually you'll get that body cell. The generative cell divides, and then one of the cells is the body cell, which divides the sperm. All right, so then the image is trying to show that. So when you have the pollen cone, and you look carefully at the pollen cone, that's microsporangium tissue, you know, in the scale area cone. And then there's a bunch of microspores in there. They all go through meiosis. All the resulting cells, one of those develop into a pollen grain. So that's, if you start thinking about how much that is, that's just like a ton of pollen in each pollen that gets made in the immature state. And then they're showing the definition of pollination here is transfer of the pollen from the male cone or pollen cone to the ovule. And right where that micropyle area is, and then they gotta grow the pollen tube down to get to the egg. But remember, it's offset, so pollination occurs, but the female's not ready yet. It's still in the megaspore mother cell stage. We've talked about the timing and how it's really weirdly offset. So you have the pollen getting transferred to the ovule, but a whole month later, you still, you know, the megaspore mother cell is going through all the process of meiosis and producing 4 cells, reduced down to just the functional, and then the functional megaspore has to develop into the megagametophyte, and that takes 13 months roughly for all of that. So the pollen's just kind of sitting there going, all right, I'm waiting. And then finally you get, after 15 months, and the egg is good to go. So a long stretch of waiting. And then eventually you'll have the sperm showing up. That takes a while, as you know, it's not immediate. Wouldn't make sense to have the sperm show up when you still have like 12 months to go for the female to get ready. Do you remember what the time range is for pollination to good to go? Maturation, everything's good? Yeah, 18 months. So 15 months, you get the sperm released. One sperm fertilizes the egg, the other one typically dies. So it's a race. One sperm makes it, the other one does not. And then you get another burst of lots of mitosis. So the pre-nuclear stage, again, that's kind of the hot mess stage where it just doesn't look like a whole lot of anything going on, but eventually it's a lot of mitosis going on. And then eventually you'll see really nice development where you get a very nice embryo, you get seeds developed, seed coat developed. So that integument tissue that's surrounding the ovule, you're going to develop a seed coat. And yeah, it takes 18 months on average for all of that to happen. So here they're showing pollination with the pollen grains coming through the micropyle. And that's the megaspore mother cell that you're looking at there. So that is this whole thing here. That's megasporangium tissue. There's no megagametophyte there yet. It's not there. There's no egg. There's no archegonia. So the pollen grains have to sit there and wait until all of that starts to functional megaspore. I mean, the megaspore mother cell does its meiosis and functional megaspore, megagametophyte. And eventually you get full fertilization, embryo development. So the embryo's in green in the middle there. That's the baby sporophyte. What's the pink tissue? We're looking at the seed. So what's the pink tissue? This is tricky. The integument became the seed coat. So the very outside of it, that's the seed coat. But this is tricky because we're in the gymnosperm world. So all this pink stuff is not endosperm. When you're in a botany class and we say endosperm, we're talking about a sperm cell fusing with the polar nuclei in a flower in the inside there. I'll show you that in a minute. That didn't happen here. So what we have going on here is going back to this. So here's the egg. And that's the megagametophyte. So once the egg is fertilized, it starts growing into the embryo, and then the baby sporophyte is also two stages here. Does all that right there. So it's still in this megagametophyte as the seed is forming. It's all right there. So when you look at a fully mature seed, the pink is megagametophyte. Okay. So this is a hard concept because in like other classes or in your normal life, you're always taught that seeds have endosperm. You know, that's just like a thing. People just always say it. It's like, well, yeah, not always. They're thinking about angiosperm. They're not. Gymnosperms get short shrift on a lot of that kind of common knowledge idea. So please don't forget, gymnosperms are special in the sense that their seeds have megagametophyte tissue for nutritional support. The sporophyte, if you call on nutrition, take that nutrition from the megagametophyte as it starts to germinate and grow. Yes.
If there's a question about that, by the diagram, but you're saying like, oh, this thing.
Yeah, I qualify it. Yeah, I qualify it. Yeah, because again, it's most people will jump immediately to the angiosperm seed idea. So I always qualify it. Look at this gymnosperm seed. What's going on here? So yeah. Let's see. Any other questions on this slide set? Or gymnosperms? See, we looked at the pollen cone already a little bit. Pollen greens. Why my computer's slow? So here, the integument surrounding the megagametocyte, that integument tissue thins out, but toughens up, becomes deep coat. And it's the megagametocyte inside the seed that serves as the nutrition source. And then cones, remember cones can seal up and open up at different stages. So it makes sense to kind of seal up during once pollination has occurred, go ahead and seal up, protect everything going on in there, and then open up again when you're ready to release the seeds, or you'll be open for pollination as well. So we looked a little bit at that when where's that picture here? This idea, closed or open. The second slide set got into the pine needles, fascicle idea. So fascicles is just the bundle of needles. Once they get more mature, first they start out where they're just single needles. They're very young. And then after a couple of years, they'll start differentiating into fascicles, which are just bundles of these leaves. So this slide 8, you're looking at 3 needles in a fascicle that were chopped in cross section. Recessed stomata. And then we kind of did a survey of the different groups, phyla, the cycads, which look like palm trees, but they're gymnosperms. And then the ginkgo, which again looks like an angiosperm, but it's really a gymnosperm, so careful about that. Only living member, Ginkgo biloba. And then the wacky looking needlephytes, where you have Phaedra, Needham, and Welwitschia. So they're kind of a wacky group. And it was in the needlephytes that we see some peculiar ideas showing up, because they have some similarities to the angiosperms, more so than the other phyla. And initially the scientists got all excited and thought, oh, maybe the neophytes are the immediate ancestor to angiosperms, but they don't do that anymore. They're like, no, guess not. So they think it was, so there was a common ancestor, and the neophytes branched off, and then the angiosperms branched off. But they're a little different. All right, should we shift to the angiosperms? Angiosperms, we have certainly the life cycle to think about. Flower anatomy, I'll be asking about flower anatomy for sure. Fruit anatomy as well. And then the polygonum type embryo development, embryo sac or megagametophyte development will be emphasized. So megaspores and microspores again. With the megaspore idea, we have the megaspore mother cell inside the megasporangium. That's one thing. That's the chromosome, and that's all megasporangium. Carefully protecting it. It's not, spore's not released. It's kept protected. And then that megaspore mother cell goes to meiosis. You get 4 cells, 3 die off. You're left with functional megaspore. Okay, it's still retained inside the megasporangium, similar to what we saw with the gymnosperms. So now that functional megaspore has to grow into the megagametophyte. But in the angiosperm world, they decide to call it the embryo sac. I'll try to use both words in the exam question. So I tend to like megagametophyte because that's what we have been doing up to this point. Irritating stuff on that, but anyways, so here we have the functional megaspore. It's only 3 rounds of mitosis, not like with the gymnosperms where you have that hot mess of tons of mitosis. Here we just have 3. They're very simplified it, and then they migrate. But you have the situation, it's a little odd because you end up with 8 nuclei, but only 7 cells. That's an important concept. And so there's 2 nuclei in that central cell, which is weird, but how they do it, at least for the polygonal. So you have that central cell, it's 2 nuclei, and then please know the anatomy of the embryo sac. So don't be surprised if you see a big one where I have nuclei, total cells, the egg, synergids, the integument. Questions on the life cycle, or the megagametophyte idea? At least it's pretty straightforward, just 3 rounds of mitosis, pretty straightforward. And then with the pollen, the microgametophyte, we're zooming into the anther. And so that's the microsporangium tissue with all the microspores. Each microspore will go through meiosis. Each resulting cell can develop into a pollen grain. But now they only do 2 cells when they're released, or the pollinator comes along and grabs them. There's only 2 cells, generative cell and 2 cells. And then you'll have pollination occurring, transfer of pollen from the anther to the stigma. And then you'll have double fertilization. So now we have the case where both sperm participate. So here, I show sperm. So they're showing one of the sperm fuses with that central cell with the polar nuclei. Now that's 3, so that's 3 aneuploidy, because you have those 2 plus the sperm nucleus, and now 3 aneuploidy. The second sperm will get to the egg. So because both sperm are involved in these fusion events, one with the egg, one with the polar nuclei, that's double fertilization. We have that right here, double fertilization. And then that central cell will keep developing into the endosperm. All right, that's where we get endosperm from. It's that central or polar cell that gets fused with sperm. So that's the endosperm. So this image, I think, does a nice job kind of clarifying that. Okay, I see we're out of time. Any last minute questions? Exam is next Tuesday, April 30th at noon in here. Yes. Yes. No,